Hello and welcome back. Today I'm going to continue talking about biotechnology by talking about farm animals. That's farm like pharmacy. These are transgenic animals that have had their genomes permanently altered through recombinant DNA technology. These are animals like transgenic sheep that carry a gene to synthesize a protein that can treat human diseases such as cystic fibrosis. Creating transgenic animals is much more complicated than creating transgenic bacteria or transgenic plants. DNA must be inserted into the nucleus of an egg or sperm, then carefully implanted into a surrogate mother. If successful, these babies will be born with one copy of the gene inserted into just one chromosome, so they're heterozygous. To create an animal that has two copies of the gene, we need to mate transgenic animals with each other and identify which children are homozygous. Once we create that animal line that's homozygous for the gene, we can mate these animals with each other and the line will carry the gene that we inserted. The most time-consuming part of creating a transgenic animal is the process of creating the initial line of animals that are homozygous. Once this is done, it's relatively inexpensive to continue the animal line. A challenge faced when creating transgenic animals is inserting the gene in the correct location. The gene needs to be inserted next to or with a strong constitutive promoter. You remember that's a promoter that's always turned on, ensuring that the gene is transcribed and translated at high levels. And if you want the gene to be expressed in, say, the milk of sheep or goat, you need to insert it next to a milk protein such as casein. So the location of the gene insertion is critical for several reasons. Remember, just like real estate, it's all about location, location, location. The types of transgenic animals that can be created are endless, but not without ethical implications. Pigs, for instance, could be genetically modified for possible human organ transplants by inserting HLA, the human leukocyte antigen. And when HLA is expressed in pigs, this will allow their tissues to be compatible matches for humans. But obviously, there's a controversy over using animals, and specifically pigs, as there could be cultural or religious issues, as well as ethical issues of having to kill a sentient animal to harvest tissues such as the heart. But by using animals to make human-compatible organisms, this would alleviate the sometimes fatal waiting game of long organ donor lists. Another interesting aspect of DNA technology is RNAi, also called RNA interference. This is when a gene is inserted backwards and makes a backwards copy of the transcript. I'll explain how this affects gene transcription and translation. The native RNA, that's the RNA that's normally found in your body that your body makes, will bind with the backwards copy of the RNA that we've created and produce a double-stranded RNA complex. This double-stranded RNA complex will be cut up by your body. That's because the cells of your body know that RNA should not be double-stranded, and it shouldn't be in your cells. So our cells have mechanisms to remove and destroy that double-stranded RNA. Scientists first observed the action of this RNAi when working with a flower called petunia. At the time, they didn't fully understand what was happening. They wanted to make a really purple flower. So they added extra copies of the gene that coded for the purple pigment. Well, in some plants, the extra purple pigment was inserted backwards, and backwards copy of the RNA transcript was made. Instead of creating an intensely super purple petunia, the researchers had flowers with patches that were white and lacked all purple pigment. How could that be if they just inserted genes to make a lot of purple pigment? The answer, as we now know, is RNAi. When this double-stranded RNA is encountered, not only do you destroy the double-stranded RNA, but our cells will go back and locate the original DNA sequence that corresponded to the RNA, and that sequence will be heavily methylated. And do you remember what happens to DNA when it's heavily methylated? The gene will no longer be expressed. We can use what we know about RNAi to stop the expression of mutated proteins that we see in certain cancers, or in the case of Huntington's disease, that's due to a mutated Huntington protein that builds up in the brain. You may have heard of gene therapy. Gene therapy works to fix defective genes by replacing them with properly functioning genes. So instead of removing gene function like with RNAi, we'll now discuss how to restore gene function. One way to do that is by using transgenic viruses that contain a properly functioning gene, along with a promoter that will be incorporated into a person's genome. This introduced gene with its promoter will enter the genome, replacing the function of a defective or missing gene. Right now, gene therapy focuses on treating diseases caused by single gene mutations. Gene therapy has been approved for treatment of lipoprotein lipase deficiency, which affects the body's ability to break down fatty acids and can cause severe pancreatitis. With all of this DNA technology, ethical questions can be raised. But most of us agree that using this technology to treat diseases seems reasonable. But should genetically engineered human growth hormone be used to stimulate growth in children who are deficient? What would you consider deficient? Should someone who's short because both her parents are short be treated with growth hormone? What about someone who makes almost no growth hormone? Where's that threshold that warrants treatment? So usually, short parents beget short children. But what if these parents just wanted a taller son? And I say son because parents are much less likely to take a short daughter to get growth hormone therapy. So should parents be allowed to use this medical treatment to help a son develop a few inches taller? The growth hormone shots can be painful, and it teaches a child that they're not good enough the way they are. Is simply being short a condition that warrants medical intervention? How do we decide what, when, and who should be treated? We can also use these technologies to diagnose disease. In 2010, a young boy, Nicholas Voelker, had a serious intestinal issue that warranted many surgeries. And the underlying cause of his intestinal condition wasn't known. So even though he underwent medical surgeries to correct the symptoms, they didn't fix the cause of his disease because doctors didn't know what the cause of his disease was. So they decided to do something extraordinary. His doctors and researchers decided to sequence Nicholas's genome. Researchers examined his genes looking for the cause of this rare disease to identify it so they could hopefully treat it. They found a mutation responsible for his intestinal disease in a gene called X-linked inhibitor of apoptosis, XIAP. He was deficient in it, so he had a point mutation. Remember we discussed point mutations in week 1 and with reference to the sickle cell trait? In this case, it was just one substitution of the 203rd amino acid of almost 500 amino acids. And the amino acid, which was supposed to be cysteine, was a completely different amino acid. Tyrosine was present, which gave the protein a completely different shape. And a different protein shape means the function of the protein was altered. XIAP has 2 important jobs. It blocks the process that makes cells die, which is called apoptosis, and it also helps prevent the immune system from attacking our intestines. In the case of this young boy, the protein had an incorrect sequence, so in his body, the immune system was attacking his intestines. This is called an autoimmune disease. When the medical team diagnosed the correct gene defect, they gave Nicholas a blood cord transplant using stem cells that matched and cured his gene defect. The last interesting question for us to consider in this talk has to deal with biotechnologies and should companies be allowed to own or patent genes? Let's examine one gene. It's called Delta 32 version CCR5. It's inherited in a simple recessive fashion. What that means is someone has to have 2 copies of Delta 32 allele in order to have none of the CCR5 protein present on their cells. And people with 2 copies of this allele are highly resistant to the common type of HIV. They can be vulnerable to other strains, but for the common HIV strain, it's very difficult for the virus to enter the cells. So a patent of the gene, the CCR5 gene, was given in 2000, and obviously there was a lot of outrage in the scientific community. Who should own the right to this gene sequence? Should anyone own it? Also back in 2000, a woman in England wanted to become the first person to patent herself. Donna McLean filed a patent for her genome stating she was novel and useful. In the United States, there's been a lot of controversy over patenting genes. BRCA, the breast cancer genes, have been in the news a lot dealing with this patent law. Patenting gene really limits the information available to researchers. And because of this, the Supreme Court ruled in 2013 that naturally occurring genes may no longer be patented. Not everyone was happy about this, as many private industries kind of hoped to cash in on that patenting.